expos, exhibitions, and conferences for imparting scientific knowledge to the stakeholders. He has worked as expert member in committees of Food Safety and Standards Authority of India, FSSAI, BIS, and EPEDA. He is regularly providing consultancies and involved in technology transfer programs for promoting entrepreneurship in mid sector. He has more than uh, 20 awards to his credit, including ICR Lal Bahadur Shastri Outstanding Young Scientist Award of National Academy of Agricultural Sciences, KU Patel Memorial Award of Indian Food Processors Association, Associateship of National Academy of Veterinary Sciences, Fellow of Indian Association of Veterinary Public Health Specialists, Fellow of Indian Meat Science Association, and member of National Academy of Science India. He has also published one patent and is a member of different scientific societies. With this, he has having many achievements, but with these few achievements, I request Dr. Girish Patil sir to present his topic on cultured meat, consumer acceptance, and ethical issues. Patil sir, please.
Yeah, thank you very much. Am I audible?
Yes, sir. Yes.
Okay, thank you, Dr. Jande sir and Dr. Vilas Vaidya for the kind introduction. The topic given to me is cultured meat. I'm just a little bit expanding it to include plant-based meat also, because this is also coming up recently, and then last week only Licious also launched plant-based meat products. So I'll be focusing and just be giving brief introduction to both cultured meat and plant-based meat as an alternative to the conventional meat. So content overview, I'll be telling a brief overview of Indian meat industry, projected demand for the meat by 2050, input constraints in enhancing the meat production, insights into cultured meat sector, and insights into plant-based meat sector, and alternative meats from the perspective of different stakeholders, and scenario analysis. Okay. So to give an overview of meat sector, in India, 71% are non-vegetarian as per the government survey. So 71% means we have around 1 billion people are consuming meat, and we are the second biggest market for the meat after China. So it's a very huge market, and meat contributes immensely for the animal protein consumption and to meet the animal protein requirements of the huge population. Meat group contributes around 2,18,000 crores to the national GDP, and the sector is growing at around 7% per annum. Okay, and the meat production in India doubled in the last 10 years. So in around 2009, around 2010, 11, we were around 4.4 million tons or so. Now we have become around 8 million tons. So the meat production has doubled in 10 years. And then we also export meat, around 1.15 million tons of worth of buffalo meat, worth 3,175 million US dollars was exported. And annually around on average around 23,000 crore rupees worth of buffalo meat is exported. And India stands fifth in the world meat production. However, if we see as a contribution, overall contribution, it stands only 3% of the world production, but we have highest number of livestock heads in the world. If you see the contribution of different species, more than half of it is contributed by poultry. So it is followed by buffalo meat. It's around 20%. But however, around 80% of that is exported. Then around 14% is goat, and around 8% is sheep, and 5% pig. Now projected demand for meat. So let us see what will be our projected population by India by 21st century. So different people have projected. So overall, we can say that our Indian population is 1.6 billion by 2050. Okay. Now we are planning for 100 years of Indian independence, 2047. So our government has given as a target of making India self-reliant and a developed country by 2047. So nutrition security is very important requirement of that. So now we have to cater to 1.6 billion population. At present we are 1.3. Okay. So apart from that, the population is increasing. Okay. Then India is also no, the per capita income is also increasing. So as per UNDP report 2019, around 271 million citizens have moved above poverty line in last 10 years. But today only at Times of India a report has come wherein it was told that in the last 15 years, around 450 million people have moved above poverty line in India. So that means the overall per capita income of the country is increasing. Now, increasing and over worldwide, it has been seen that with increasing population and with increasing per capita income, demand for the meat and meat products also rises. So that is one of the reason why in the last 10 years meat production has doubled. Means during that period there was an addition of population. At the same time, income of the population has also increased. So with increasing population and demand, the demand, as you can see in this graph, overall in the countries which consume very high meat, so where with increasing per capita, higher the per capita income, higher the consumption of meat. So similarly, in the next 25 years, the country will grow. Once the country will grow, its per capita income will grow, and demand for the meat and meat products will also rise. Okay. Now, as per the FAO, so it was told that it will be 10 tons, by but even this 18 million tons also appears to be on the lower side, because now we have already reached around 9 million tons, 8.6 million tons. So by 2050, we feel that at least our meat production should be tripled. At least 27 million tons we should reach it. Okay. Now that is the requirement. Let us see the input constraints in enhancing the meat production. So because whatever the quantity or for animal livestock product, we require inputs. So main input required is water. Okay, so water is the main input. According to the Composite Water Management Index report released by Niti Aayog in 2018, 21 major cities are racing towards the ground zero. Okay, and the report also says by 2030 the water demand is projected to be twice the available supply. Okay, so whatever in the supply, it will be twice. Okay, so implying severe water scarcity for hundreds of millions of people, and in eventual 6% loss in the country's GDP. So recently, two places I visited. One in somewhere in Karnataka, a college, wherein they have a dairy farm, but in the summer they cannot manage it because they are buying the water, because even for the routine guest house and quarters or the hostel and all, they are buying the water. That is so how the scenario is changing. And also I recently visited in one institute in Nagaland. So I reached by afternoon, and I was horrified to see that there is no water in the guest house. And they say that every day they are getting only 1 hour water, and very limited way they are using it. So that is the scenario. Slowly, yeah, year on year, we are seeing this even in Chennai also. So water was to be brought on train. Sometimes there was a flood, and sometimes there was a severe water scarcity, forcing them to bring the water by train. So these are all impact of the climate change and excessive use of water. Our groundwater level is going down. So this we have to keep it in mind that the water, so demand for water is increasing, whereas availability is decreasing. And so the demand will be 2 times the availability by 2030. So that is what our Niti Aayog says. As per the CEO of Niti Aayog, so India is currently suffering from the worst water crisis in the history. Okay, so you can see that in the map that the states in the red, they are facing severe, extremely high water scarcity. They are facing it. And what will be the impact? So once the water level comes down, so the first priority will be for the government will be providing the drinking water and household requirement, and then comes agriculture, and then comes industries. In that order it will put. So that means the water intensive industries such as food and beverages, textiles, paper products are likely to be worst affected. Now as of now we are getting ad libitum water, in our house or in our farm. Now, assuming that the days are not far off, wherein meters will be put onto the, our tap, how much you use, and just like electricity. So, as much as you use, that much you have to pay. If those days come and they are not far off, will we be able to give ad libitum water to our animals? Will we be able to maintain large, livestock population what we are having now? So that is the question. These are the sustainability issues. And another important is feed. One is water, another is feed. Animal feed availability scenario in the country. So as per the Parliamentary Standing Committee on Agriculture, so there is a scarcity of all three types of feed resources. There is a dry feed, green feed, or concentrates. Okay. So by 2025, it is estimated that dry feed will be short by 21%, greens by 40%, and concentrate by 38%. This is a level, means on an average. So we are deficit by around 25%. And also Department of Animal Husbandry in the year 2019, it has given advisory, it has issued advisory wherein it has showed while the number of livestock is growing rapidly, the grazing lands are gradually diminishing due to pressure on land for agricultural and non-agricultural uses. Most of the grazing lands have either been degraded or encroached upon, restricting its availability for grazing. The area under fodder cultivation is limited to about 4% of the cropping area, and it has remained static for the last 4 decades. So in the last 4 decades, the grazing land is static for 4%, and we cannot expect that in the next coming 3 decades it will increase. It will only reduce. So this is the input scenario. So as per a report of Indian Grassland and Fodder Research Institute, okay, they have mapped different states with scarcity. So you can see that the states which are colored in green, they are surplus. So very few states are in surplus now. Whereas the states which are in red, they are facing fodder scarcity of more than 50%. Okay. Whereas the light red, it's around 25 to 50%. That means most of our area, so they are under, we are facing a scarcity of availability of the fodder. So this we have to keep it in mind. And the one more issue is global greenhouse gas emission as a as when we say Vasudhaiva Kutumbakam, so every country has responsibility to reduce the greenhouse gas emissions. So among the greenhouse gas emission, it has been found that 13% is from the agricultural production. Out of this 13% from the agricultural production, enteric fermentation. Okay, so this is the thing. So we can we cannot go on saying that ours is lesser and industrial production is more, vehicle pollution is more, and all these things are there. The thing is, now United Nations has told individual action. No need to look here and there. Whatever possible to reduce the methane emission from into the into the atmosphere, we should do it. In our sector, whatever possible, if there is any possibility of bringing down the greenhouse gases, we should do it. That is one of the reason why some of the countries have put restrictions on consumption of meat and rearing of the livestock. Some of the countries have already put because the cost of this climate change, so they are unimaginable actually. The what we are saying is really unimaginable thing, because the kind of heavy uneven rainfall, and sometimes scarcity, sometimes excess. These are all impact of the climate change, and we have the responsibility to do in whatever the form we have. We should reduce the greenhouse gas emission from the to the atmosphere. Now the population growth, increasing demand for the meat, resource constraints, and also they are taking they have given because of this, people are moving towards the alternative meats. Okay, so alternative meats, in these two things come. One is plant-based meat, another is cultured meat. These are the two options they are seeing. Even some of the visionaries like Bill Gates and Richard Branson, they have told, Bill Gates had told there is no way to produce enough meat for 9 billion people, yet we can't ask everyone to become vegetarians. That's why we need more options for producing meat without depleting our resources. And then Brosnan, he also says that I believe that in 30 years or so, we will have no longer be we will no longer need to kill the animals, and that all meat will be either be clean or plant based, taste the same, and also be much healthier forever. So it is not that whatever they say is the truth, or the gospel truth. So these are the businessmen. So whatever the business they have done, if you see the past 3 to 4 decades, these are the leaders, business leaders. They have started something which will come in the next 30 years. So they, whatever the Google does, Google whatever they do, they will think what is required by 2030 or 2040 or 2050, and now they start working on it. Similarly, these Bill Gates, Branson, Sundar Pichai, they have all invested in cultured meat. They feel that there is a huge scope, and these cultured meat and plant-based meats are, coming of age, and they are the future. That means we can take them as an indicators that these things are coming up, and India cannot shy away, and we cannot simply ignore these developments. Now coming to the insights into cultured meat. So first the concept, the cultured meat. Let us understand what is cultured meat. Here, uh, basically, now what is the normal production? The production of meat. How do you do? We bring the animals, we rear it. Hello.
Sir, yes, you are audible, sir. Please continue.
Okay, okay. So we rear it and we slaughter it and we produce the meat, and from there we make the processed meat products. Whereas in cultured meat, so you take a biopsy or take few cells, isolate the stem cells, multiply and produce the product. This is an alternative way of producing the meat. Okay. So now how this idea came in the year 1931, in another visionary Winston Churchill, he wrote, "We shall escape the absurdity of growing whole chicken in order to eat breast or wing by growing these parts separately under suitable medium." I know he told by next 50 years by 1980. But it took more than 50 years. In the year 2013, it took 20 years more. On 5th August 2013, Dutch scientist called Mark Post, he produced the first lab-grown burger, and he has presented and he demonstrated before the media, and that created the huge interest, in cultured meat. So, and then let us go in deep into this sector. What is lab grown meat? Lab grown meat is the meat produced by in vitro cultivation of animal cells rather than slaughtering of the meat animals. So, this technology allows meat to be cultured from cells in a fermenter or bioreactor. So, how do we multiply the cells? We go for large scale multiplying of the cells in a bioreactor. So, what we do, we take a biopsy or extract the stem cells from the embryo, okay, and then we characterize it, and then we have to, there are certain other aspects to be standardized, and then we put the cells into the bioreactor. We provide them the support called as scaffolds. So from there the cells will multiply. So the large scale multiplied cells, they're put in the format and the products are produced. Okay. Now what are the advantages? So there are different varied kinds of publications. You can see some people say that it is not at all beneficial. Some people say it is highly beneficial. But it is understood that if you are able to do it at the lab scale, so then it will definitely, it will use less land, less water and less energy. So that is for sure. And what are the challenges in the cultured meat production? The first thing is we should have the cell lines. Then we should have the proper media. Okay, the proper media in which these cells can multiply. Then we have to provide the scaffolds. Okay, because myoblasts are attachment dependent, we have to provide the scaffolds so that we can provide proper shape to it. And then we have to upscale it in a reactor, and then it has to meet the organoleptic properties of that of traditional meat. So these are the 5 challenges of the cultured meat. As of now, 4 countries have already achieved cultured meat production. The first was in Netherlands, I have already told, and then Israel has done it, and then South Africa, and then China, okay, and all these countries, and then Singapore. These are the 6 countries rather. In Singapore, cultured meat is available already in the commercial scale. Coming to the first challenge, isolation, cultivation, and differentiation of the muscle and other stem cells. So, what are the sources of cell? Embryonic stem cells, myosatellite cells, induced pluripotent stem cells, and adipose tissue derived other stem cells. So, all different stem cells. Why? Because these stem cells they multiply, more number of times. If you take, adult cells, they will not multiply because they have already reached the senescence. That is why we have to go for embryonic stem cells are the best. Okay, they will multiply more. Even then, to achieve continuous multiplication, some people are also going for the genetic modification, includes the TERT, to make it the pluripotent stem cell, induced pluripotent stem cells. So cells should have the capabilities to proliferate indefinitely, embed in a three-dimensional matrix, and conditioned to get the mature muscle fibers. So in the year 1963 itself, Koenigsberg, he demonstrated the existence of muscle stem cells from day 11 to 15 chicken embryo cell, which can readily proliferate 20 cell doublings. So now it has been recently found that if you are taking the embryonic cells, they can even go up to more than 27, even up to 50 to 60 doublings is also possible. Okay. Now, the natural doubling level of chicken muscle stem cells is higher as compared to the somatic cells, which have a limit of 40 to 60. Okay. So now, generally, there is a term called Hayflick limit. Hayflick limit means after certain multiplication, after certain divisions, cells will cease to multiply. That is called Hayflick limit. So in order to overcome this Hayflick limit, either we have to go for embryonic cells or we have to go for genetic modification. These are two options to develop the cells. So how genetic modification is done? So telomerase activity restoration is an ideal method to enhance the prolific cells. So what will happen? No, these chromosomes, as the number of multiplications and number of divisions increase, the size of the chromosomes will go on reducing. Okay, and at one particular time it will stop multiplying. So that can be inhibited by inserting chicken telomerase reverse transcriptase. So if you put the telomerase reverse transcriptase, if we are putting in those cells, then that shortening of the chromosome will stop, and then cells will go for more doublings. And one more challenge is co-culturing, because now we have, it is not just myoblast, but we need myoblast, fibroblast, and adipoblast. We have to grow them together. There are different co-culturing techniques are there. Okay. So this co-culturing simultaneous multiplication of different cells in the common media, or multiply them separately, or then mix them. That is also a challenge. And then I am not going into these brief other stem cells. And second challenge is selection of media, which is cheaper and free from the animal sources. Now, just like the animal livestock sector, where 70% of the input cost of an livestock farm is by feeding. Similarly, 70 to 80% of the cost of production of the cultured meat by is because of the media only. Obviously, it is by media. So for that purpose, we should have some media which costs less, and we should have the media which is animal derivative free, because we are claiming that it is a animal, we are claiming that traditional meat products are not good and all those things, means that is the one of the reason why people are going for cultured meat. So here, that is why if you use the serum, because fetal bovine serum is the one component which is used for all cell cultures. Okay, by multiplying the cells by fetal bovine serum, it will not serve the purpose. So that is why we have to replace the fetal bovine serum, and several strategies have been followed, and we are also working on it. Okay, and certain commercially available serum free media are also available: ultraser, mushroom extract, cyanobacteria. The problem is most of these serum free media, they are standardized mainly for the human cells, or the rat or hamsters, whatever is generally used for the in the pharma industry. Because but for these mammalian cells like livestock cells like chicken, or maybe for sheep and goat and all those things, these things will not work. So we need to standardize ourselves. So there is one more called E8 medium, essentially 8 medium. It is a combination of basal medium, vitamin C, sodium selenite, insulin, transferrin. So what they do, in the serum certain growth factors are there. Instead of putting the serum, growth factors are being expressed separately, and then they are added into the medium. That is also another approach. So now, in the January 2022, the same Mark Post, who first started this cultured meat, he has also published a serum-free media formulation for cultured meat production, which supports bovine satellite cell differentiation in the absence of serum starvation. So he has reported. So like this, several serum-free media's options are coming, and after among those options, we have to find something which costs lowest, and then we should work on it. The third challenge is scaffolds. Selection of the suitable scaffolding for providing the 3D structure. Because when we are culturing, it becomes a mass. It's a pellet when we are doing it. So, but we have to provide a shape. For that purpose, we have to require a scaffold. So, myoblasts are attachment dependent, so require substratum or a scaffold. The scaffold must be edible, biodegradable, derived from non-animal sources, and must have ability to stretch mechanically. So, what are the examples of scaffolds? Chitosan, alginate, cellulose, collagen meshwork, collagen spears. So many different options are there. Nowadays, people are also coming out with edible scaffolds, so that there is no requirement of separation of the cells from the scaffolds. So what are the other benefits of scaffolds? It stimulates the fiber and meat texture, and it will helps in molding the shape in millimeter or the bigger scale. That's the thing. And fourth challenge is upscaling of culture to produce the meat. Now, upscaling means we require a bioreactor. Nowadays, bioreactors are generally used for, we have a system called fermenters, wherein the bacterial cells are under anaerobic environment are multiplied in large scale for production of the vaccines and different purposes. Okay. But what we require is, we don't require a fermenter, we require a bioreactor. Fermenter works under anaerobic condition, whereas bioreactor works under both aerobic and anaerobic. So we require aerobic, we require a bioreactor which can provide oxygen to the cells. So now, what are the things to be maintained in the bioreactor? Continuous suspension of cells, high output, contamination-free operation, large surface area, low shear and uniform perfusion, and supply of oxygen and remove the carbon dioxide. Now, types of bioreactors: direct perfusion bioreactor, okay, rotating bioreactor, different things are there. So here basically we have to provide the oxygen, whatever the carbon dioxide is produced and whatever the excreta of the cells are there, they are to be removed, and we have to provide the proper environment so that the cells will keep multiplying, you know. And now efficiency of evaluation can be measured by milligram of product produced per liter per day. That is how we calculate the efficiency of this. Then final thing is acceptability. So whatever we produce, unless and until it gives the required taste, people will not accept it. So it has to meet the composition, texture, color, sensory acceptability, and all those things. Okay. So color, consistency, saltiness, tenderness, moisture, vitamins, all these things are important. So how people achieve this? Once they multiply the cells, they add non-meat ingredients so that it gets into different required texture, juiciness, consistency, and all those things. Okay. Now, co-culture of myoblasts, embryonic fibroblasts, and endothelial cells. So this will help in co-culturing. Will to certain extent will help in reaching these required, you know, these acceptability parameters. Okay, sensory parameters. Now, we at NRCN Meet, in collaboration with the CCMB, we started a DBT funded project called Enabling Technology in Mammalian Cell Culture towards Cultured Meat. So we have made rapid progress in this, in serum free media, and then now we have ECM extraction of the cell, isolation of cells, and all those things we have made. And now what we have made is we have joined hands with a firm. Okay, that I will explain. So in different newspapers also it came. So in the last 5 years, the number of companies who are venturing into lab grown meat, it is increasing rapidly. Okay, by 2020 around 60 companies, now around 70 companies across the world are working towards it. Okay, they're putting a lot of different technologies they're bringing out, and where however US, Singapore, and Israel, they are leading in this cultured meat research. And after making initial progress in this project, we have joined with a company called Neat Meat. It is based in New Delhi. So they are funding our project, and we have taken the project under the contract research mode. So just 2 days before, an article also came in the Hindu regarding this. So now they are funding us. Yesterday only we are happy to share that we got a seed funding from one of the sources around 30 lakhs. Today also we have a presentation in DBT to get more funds for it. So now with the private industry partnership, because some of the projects, by going with the government funding is very difficult, because we have constraints of the procurement and having good manpower and all those things. That is why, where if you go with the industry, then if you want certain chemicals, we can directly go and buy it. So we got a good funding, industry also. We have joined with the Neat Meat, and we have made good progress in this direction. CCMB is also with us in this. So what are the prospects of cultured meat? If you see, for Indian conditions, beef has the least prospect, because the consumption of there are not many beef consumers in India. So buffalo meat is also not much prospects, because buffalo meat is mainly exported, not many consumers in India. Whereas mutton is a bit higher, because mutton and chavan, they are generally small ruminants. They are depending on the grazing. These grazing lands are coming down. So that is why there is a good scope for going for mutton and chavan, because whatever we produce, the meat from the small, that's now it has some of the places the price of the chavan has gone up to 900 to 1000 rupees also. So rapidly there is a huge gap in the demand and the supply. So highest prospects they compete with the human food. They take the maize. It is completely concentrate based, because there is no grazing, there is no utilization of agricultural byproducts. Because cattle, buffalo, sheep, goat, to some extent they use the agricultural byproducts and they convert it into meat. Whereas chicken, it is completely dependent on this. And also demand for the chicken is rapidly rising. As you have seen that in the last three decades, it has rapidly grown. Now half of the meat consumed is chicken. So there is so more sources of production of the chicken can also be included. Now, Singapore, as I told, Singapore is the first in the world to approve the lab grown meat for sale, because although technology was there, So, no permission getting, regulatory clearances was an issue. So in 2020, Singapore Food Agency, after doing all, necessary safety studies, they have permitted. Okay. And now because of this permission, restaurant they are selling it. So in 2021, so around 20 companies from across the world, across the world, they are putting their base in Singapore because there is a necessary regulatory environment required for moving towards the cultured meat. Okay. And some of the people who have tasted say the nuggets are a little spongier than the regular chicken, but crispy and delicious, just like the real thing. Slowly these things are coming up. So I think this week only, US FDA has cleared the guidelines for the cultured meat because one of the main problem was FDA guidelines were not finalized. Now guidelines have been finalized and they have been notified. So once US comes up, there is a huge, I think progress in this sector is expected. Now, regarding the price of cultured meat, the fastest tasting of cultured meat in 2015, they say that it was around $330,000, per grown, per burger. Whereas future meats, they have brought down the cost to $1.7 per breast because a lot of research is going on to bring down the cost of the media. Okay, and acceptability. We did a survey and we found that more than half of the Indian consumers they want to taste, they want to try it. Regarding the regulatory landscape, FSSI, we have a category called non-specified foods. So if we have, as of now, there are no exclusive standards for the cultured meat. If someone wants to market it in India, they can submit their application under the non-specified foods. Okay. So now there is no competition of meat industry and meat alternatives because the population is increasing, income is increasing, cultured meat, animal derived meat, and plant-based meat all together they should, I think by 2050, all of them will be part of our food basket. Now let us give some insights into the plant-based meat sector. What are plant-based meat? Plant-based meat products are made to mimic the properties found within the natural meats. Okay. Now the process of creating plant-based meat product involves protein sourcing, isolation, functionalization, formulation, processing, and distribution. Now, a survey has showed that as compared to 2015, in 2019, there was around 600% increase in availability of the plant-based burgers, around 400% availability of plant-based sausages. Now, in India also, whether you can see all these Licious, Swiggy, Zomato, they are also selling these plant-based meat products. So several companies they are coming up. Okay. And now there are the research into plant-based means alternative to meat was going on since many centuries. In 965 AD itself, people tried with the tofu and then the various experiments, and finally in the 2014, Beyond Meat launched breast burger. So that Beyond Meat and Impossible Foods. So they made the huge impact, and then all these plant-based meats are available in the supermarket shelves of all developed countries, Europe and all those things. Now slowly it is coming into India also. So what are the basic ingredients which are used for production of the plant-based meats? For the protein purpose, soya protein, pea protein, soya beans, hydrolyzed vegetable protein, mycoprotein and almonds. And for the fat, vegetable oil, canola oil, sunflower oil and all those things. And carbohydrates, potatoes, tapioca. And fibers, different things, and vitamins and minerals. The color of the meat is achieved by using the red beet juice. Even for the cultured meat also, when we are culturing the cells, okay, so that will not have any color. It will be transparent. That means it is a colorless. So to get the color, they go for adding red beet juice and different colorants they add it. Actually. Okay, this is beyond sausage and beyond burgers. They're all there. A nutritional comparison. So now what they're claiming is the plant-based meats, they have the zero cholesterol, okay, and the least fat. And however, in terms of bioavailability, the animal proteins are always better. There is no doubt in that. Animal proteins will, because it meets all the nutritional, all the required amino acids, essential amino acids will be providing. Bioavailability in the animal protein will be better. But however, plant-based meat people, they say that it's having lesser cholesterol, lesser fat, and more of a protein like that. Okay. And then this is a comparison of energy, protein, fats of the plant-based burgers, meat burgers. The different articles are there. Now there was a report of Barbas in 2019. They say that food sources in their natural state contain thousands of compounds that are capable of impacting human health. The vast majority are not appearing on the consumer nutritional fact panels. As a whole, foods contain hundreds. What they say is it can never replace it. It's only one more product on the menu, but the what the meat gives in terms of the nutrition, it is not just the protein, fat, and ash or minerals, vitamins. There are so many millions of other compounds which helps in health. So to some extent, it's only another product in the menu. Some vegetarians who want to go for the meat, they can go for the plant-based meat, but nutritionally, it can never replace the meat. That's the thing. So what are the investment in the alternate sector? Investment in the alternate meat sector surged in 2020, hitting almost $3 billion. Okay, it is three times the amount of the capital they raised in the 2019. And the Aleph forms and different companies are coming out with different fundings. And then which are the plant-based companies in India? Uh, so we have we have Richard Brosnan, who has invested in the Upside Foods, Emphasis Foods. We have Cargill, we have Future Ventures, Threshold Ventures, and then so all, as I told, Richard Brosnan, Bill Gates, they have all invested in this. And recently, Nestle, largest food processor in the world, they have joined hands with the Future Meat Technologies of Israel for production of the plant-based meats. Okay, Nestle, part of Israeli startup to make the plant-based meat products. Okay, and Impossible Foods has joined hands with the Google venture to get all the required investment. Okay. In India, Indian plant-based meat companies are Good Dot, Vegeta Gold, and Veggie Champ, Wesley, and last week only another Shakahari. So many others. We are every month one new company is coming and putting their hand into it. And recently, so these two film stars, they have started Imagine Meat. Okay, so this is also a brand of plant-based meat. Ritesh Deshmukh and Genelia, they are also aggressively going into this plant-based meat. And to give an example of the Beyond Meat, the company was founded in California-based entrepreneur Ethan Brown in 2009. The company received venture funding from Bill Gates. Okay, the company began selling its plant-based chicken products. Okay, in 2013, they started just around 9 years before. Then Tyson Foods purchased 5% of the stake in the Beyond Meat. You know, Tyson Foods, they are the major meat processors. They also bought the venture, invested in it. Now the company now claims around 27,000 different points of distribution for their products in the US and was rolling out 50 international markets. And as of July 2019, Beyond Meat has a market value of $11 billion. In 2020, Beyond Meat launched an e-commerce site also for selling this. So, now don't think that these they are far off. It's not that it's not unreachable like that. We should not think, because KFC also rolled out plant-based fried chicken with Beyond Meat. They have collaborated with the Beyond Meat. They're also coming out with the product range. It is made from the plant-based meat. Even McDonald's also coming out with the plant-based commodities. So in the year 2018, a Champions of the Earth award was given by UN, wherein Impossible Foods and Beyond Meat, they were awarded. In that list, we also had our Prime Minister Narendra Modi ji for policy leadership. So to that extent, they have made the progress in the area of, and they are showing the promise to the people also. It is not that whatever the plant-based options are happening, it's only with the meat. We have plant-based dairy products and plant-based egg products, plant-based fish products. Recently, FSSAI was to come out with a notification saying that whatever produced no by the source other than the animals, we cannot label it as a milk product or dairy product. So now slowly people are changing all these labels, but they are coming for these plant-based items are coming for all animal-derived products, even fish also they are coming. Alternative meats from the perspective of different stakeholders. So for the processors, cultured meat open up the new business opportunity. We get huge number of calls. There are so many investors and there are so many startups nowadays. New culture has come, startups. So they want to invest in this and they want to make the business opportunity out of it. And for consumers, one more option on the menu. Okay. Policymakers, they need to regulate the production methodology, because now even for the plant-based meats also already communication has gone to FSSAI why they should not put the word meat. They can put something else. So now these policy issues are to be addressed. Then meat handlers will get the new opportunity in plants of producing cultured meat. For the research and academians, huge scope for research and development and for sharing the global development. See, now it's very, very important because most of the times what happens now in India, we keep waiting till somebody else does it. So once some other country make it and they make commercialize and then we start, by that time it will be too late. So that is why researchers must step in, because this is a very huge business opportunity and huge export opportunities also. So we should not lag behind in this area of research. And farmers, India is an agrarian country. So major chunk of the population is in the rural areas. So what will happen to them? That is one of the big issue. But the thing is, the way things are going, we these alternate meats will not replace whatever the livestock sector. In view of shrinking resources, in view of increasing demand, so all possible resources of meat productions are to be brought in place so that country becomes nutrition secure in the by 2047. So scenario analysis of alternative meat sector. Now what is happening is, no, India, if India closes its sites and applies restrictions on meat alternatives expecting threat to farmer. Some people are already telling, what is this nonsense? So this will all affect our farmers, and all those things. Now, if we do that, so there is one something called product adoption and product life cycle. So now some people, in the stage one, innovators, stage one they go, like countries like Israel, Singapore. Whenever some innovation is the first stage, they go and they make business out of it. And early adopters, and then we have early majority, we have late majority and laggards. In most of the time in India, we are laggards, because even some of the very simple apps, India is a IT hub, but even WhatsApp, a kind of platform like WhatsApp also, we could not make it in advance. Someone made. Now WhatsApp, something it is managed from US, and all of us we are happy using it. We never think that we should develop something. Even if we develop now, it cannot be adopted because people are already used with the WhatsApp. Now so many other things have come like Signal and Telegram and all those things in some of the groups in our own Indian Merchants Association. People thought, why this, let us go to Telegram. But in spite of our best effort, we could not make, because now for social media, people are used to WhatsApp. So once the people are used to it, getting into the market is very, very difficult. And even this is whether it's a Zoom or app also what we use and all those things, we don't have anything from India. So whatever we use, it is from other countries, and we use, and they make huge profits and they take huge revenue. So that is why. So now this is a stage. Now it has already innovation stage is gone. It's already early adoption stage has started. So India, if it now also if it closes, India will lose the early more advantage. Imported products will be available in the market and will be net importing country, because we cannot stop. So once the products are producing, there are consumers who like this. So they will whatever the plant-based meat companies who are seeing in India, most of them they have a partnership with the international brands. So they take the technology from there. Here simply they produce, just like how KFC makes it. All the technology is from somewhere else. Simply we mix, we assemble and give to the consumer. Similarly, same way plant-based meat. So in our the research, the serious research into this area is not happening. Next scenario, India shows indifference and leaves it to the industry. Multinational companies will have their own plants and collaborate with the local companies to market their products. Even that Imagine Meat is also a collaboration with the international partners. And scenario 3, India aggressively pursues alternative meats with budgetary support and encouraging collaborations. India can emerge as a supplier of the world, earning the huge foreign exchange, creating business opportunities and create employment. Because now an environment of startup has come, wherein a lot of support we are getting. And recently, as I told, we have also collaborated with an industry partner, and we are getting calls from several places, and we got already we got one funding yesterday, and today also we have a presentation. Slowly, I think there is a environment of innovation is coming up, and hopefully that environment will help researchers to move and make progress in this. And next scenario is products available in India but are costlier than the traditional products, because India cost is a main factor. Whatever you say, what is its price? So products will be available with few. If it is very costly, products will be available with few takers. Product will cater to the niche market as another option in the menu. Okay. And then if there is another scenario when products available in India and are cheaper than the traditional products but do not match sensorially and nutritionally, okay, will remain a niche product, because in India they are very much particular about the taste. Even for this what you call native chicken, there is a huge demand. Scientifically, we analyzed, we could not find anything, but somehow taste-wise they like that native chicken. So they pay high and they take it. Some people don't like broiler. All the time they go for it. So taste is very, very important. And the last scenario, products available in India are cheaper than the traditional products and match sensory and nutritionally. It's highly unlikely, I feel, but Indian meat industry will face serious competition. But no, we should be prepared for the worst. Things are not far away because there were several other products which are coming from the livestock sector. Earlier, all these pharmaceutical byproducts like insulin was taken from the animals, okay, and several pharmaceutical no here what is that iron supplement, it was made from the animal blood and all. Now all those things are gone. Everything is recombinant they are making. So nowadays no takers for byproducts of livestock. All of it is made by recombinant in the lab only they are making. And next came the issue of the skin and hide. Earlier, sheep and goat skin it used to go for 300 to 400 rupees, and cattle hide are to around 3 to 4,000 rupees. Now, no takers for sheep and goat. Okay. Now, cattle and buffalo hide price also have come down by half of it. Now, only 1,000 to 1,500 rupees. The reason already there is a synthetic leather has come. They call it as a Chinese leather, which feels and all texture and its strength, everything is it is similar to that of the livestock leather. Okay, so that is why slowly that is also leather also slowly going. So these are not far off wherein all these alternate thing if they make because people are aggressively going into it something. So you should be prepared for that scenario also. Okay, the conclusions: plant-based meat alternative should be treated as a meat alternative in terms of sensory properties, but not as a true replacement in terms of nutrition. Plant-based meats nutritionally it cannot match the uh, animal meat. Then plant-based meat can be variety product on the menu at best. These products hold potential to convert vegetarians into non-vegetarians. Cultured meat holds potential to match the meat nutritionally and sensorially. Because so many changes we can make and it is possible because ultimately they are cells. But more research is required in this area, and India must closely follow these developments and explore all possibilities to capture the business potential of this latest concept. As I already told, our food basket of 2050 will have all cultured meat, animal derived meat, plant based, all will be there together to achieve the nutritional security. Okay. Thank you so much.
Thank you very much, sir, for your wonderful information on the cultured meat and plant-based meat products, the different current scenario, and you have highlighted the challenges of for the of cultured meat as well as prospects of cultured meat. Thank you once again for your excellent talk and updated information. Now I request the participants, please you can ask the questions.
Somebody is writing, if we consume a long time, will there be any health risk? Because whenever, as I told, non-specified food, before FSSAI clears any of these products, the series of safety studies are done, so including lab animal feeding and other things. So it should not cause because ultimately they have to prove that there are no chemical residues, it is all health free and all those things. That is one thing. And will cultured meat need hormones to sustain the cell growth? No hormones. That's what I'm telling. They are called growth factors, which are derived from the serum. Okay. So now serum is the source of everything. So now people are slowly going away from the serum. Serum replacers are coming. If you are using serum replacers, none of these animal type of hormone, nothing will be involved. But however, no hormones are added directly. Which type of media required for in vitro meat in culture? Okay. What type of media? So now the thing is, for cells, you are providing the environment similar to that of the livestock. You know. So the basic medium, what is it? DMEM. Okay. So DMEM is the media is the main thing, and then they add some other things, and then they add serum, fetal bovine serum. There is a combination is there. We can share with you. And these cell culture already established methodologies are there. Only thing is what we have to make is we have to replace the serum so that it makes it animal derivative. Is there any scope for flavor enhancer also? Why not? Because whatever the cells we get, they are bland because it will not have its own taste. So for getting that, we need to add all type of flavor enhancers. As I told, non-meat ingredient, lot of non-meat ingredients they add to give the similar texture and flavor as that of the meat. Okay. Really, thank you. Thank you so much.
Thank you, Dr. Girish.
Thank you, sir. Thank you.
Besides your topic, you have covered one more topic in that plant-based meat. And thank you so much for that, contributing your experience and highlighting all the aspects of the cultured meat and how it comes and what are the agencies which are mainly involved and what are the research institutions in India also involved in this kind of research in India. So thank you so much, Dr. Girish, and we'll hope in near future also you will contribute your experience in other aspects of the food safety.
Thank you,
Thank you.
sir. Thank you. We appreciate the activities of NAHEP project our department. I'm seeing since last 2 years you are organizing trainings in varied kind of programs wherein people can join online. Hundreds of people they're joining and they're getting state of the latest knowledge they're getting.
Yes, yes, yes.
Thank you so much. There is one more last question they have written. How can we differentiate? So differentiation is definitely an issue, and for this purpose, more research is required because now whether a mass of cell derived from chicken and meat itself, because there is no way of differentiation. So these issues will come. So in this area, I think people can start working also. Okay. Thank you, sir. Thank you so much, sir.
Okay, thank you so
Thanks for
much.
the
Thank you.
opportunity. Thank you, Jande sir. Thank you, Dr. Vaidya, and all the participants.
Thank you, thank you.
Thank you.
Okay, so we'll move on to the next speaker. And then Dr. B.B. Naik, sir, is a principal scientist and head Fisheries Resources Harvest and Post-Harvest Division, ICR Central Institute of Fishery Education, Mumbai. And he will speak on emerging food safety issues in aquatic food. So you just wait for 1 or 2 minutes. Sir is joining. I sharing his PPT. And Post-Harvest Division, ICR CFA, Mumbai. So to the knowledge of all the participants, I have pleasure to introduce Dr. B.B. Naik, sir. He is a Bachelor of Fishery Sciences from Bhubaneswar and Master of Fishery Science, US Bangalore, PhD in Fishery Microbiology, US Bangalore again, and postdoc in University of Tokyo, Japan. And he is the first ranker in Masters gold medal in PhD. So his education background is very, very high standard. He has handled about 22 numbers of the external funded projects. Out of that, 13 he was a PI, and the total cost is about 2,511. That is 25 crores of rupees he has generated for his institution. He has at his credit 179 peer-reviewed publications. Out of that, 114 national foreign journals. Citation is about 1590 and Nature Links is 22. He is the best research paper awardees of CPA in 2011, 17 and 21. He is he has developed various technologies. Out of that, one has granted a patent, one technology has commercialized, and one is undergoing industry validation for and four for are with ITMU. Sir is handled so many the research.
Maybe. Thank you. Thank you. Maybe we will cut short. So
Okay, sir.
thank you. I think it's very kind words. Just to tell that I have a background of microbiology and been working here in post-harvest technology division, and we are doing well as a group. My team is doing quite well. So my opportunity to talk to each one of you. Thank you. Thank you very much for these kind words. So if you permit me, then I will continue.
Yes, sir. Yes, sir. Yes, sir. You can.
Okay, thank you. Thank you very much. So first of all, my sincere thanks to each one of you for attending this, and especially the organizers for choosing me as one of the speaker for 1 hour. So I'm happy that from fisheries background, I would be able to expose the participants into the arena of microbes, I mean different kinds of microbes that are posing risk to human through various marine foods. So just if you permit me, just what is the background? There are students, there are researchers, and there are lecturers, some lecturers. Is it that okay? Am I?
Please share, sir. Share your
Yeah,
slide.
thank you. I'll share my slides. Just, I think. Okay, is it visible now?
Yes, it's visible. PowerPoint mode. Yes, yes, sir. Yes.
Okay, okay.
Continue. Yeah.
So I'm sure this program today is the last day. The participants have gone through various kinds of, I think, microbes and relationship with food and different kind of risk associated with that through the One Health concept. So basically, what we would be looking today is the water side. Say, unlike land and terrestrial animals, the fishes are exposed to the environment directly. Say, for example, that food is also coming directly in contact with the environment. So it comes in contact with the microbes, which are all already present in the water. And the fecal matter also comes back to the water. So this is why the fishes are exposed to much more types of hazards. I think my microorganisms and risks compared to what we see in land animals. Land animal, you can have a completely controlled conditions. In natural environment, it's very, very difficult to control the fishes. And when we say fishes, we have different kinds of, we actually mean fishes that are fin fishes, there are shell fishes, and there are also mollusks. Mollusks are also kind of shell fishes, but they are all bivalves. So you have the type of hazards associated with each one of them is completely different depending on their environment. Second important thing is that, just if I have to put it this way, uh, just the means of entry of pathogen is only through the food, through interterrestrial animal. Of course, through airborne and other things also could be there, but mostly it is very, very intimate in case of aquatic animal. When they respire, they have to take the water into their gill and exchange, so they take the microbes in. When they are eating, yes, they are, and the microbes are also associated in that environment, which can penetrate through the skin. And if you see the means of removal, you have very specific immune system in case of animals, and fishes, of course, have some specific immune system, not as developed or not as studied as the land animals. But the invertebrates, especially the crustaceans and mollusks, they have non-specific immune system. The means of selection and adaptation to the body usually homeotherms are the terrestrial animals. But in case of fish, the moment the temperature changes, the body temperature of the fishes also change. So that means to say the microbes have to adapt to that condition to which the fish is adapted. So they don't maintain their body temperature. Then accumulation of microbes do not happen so commonly in case of terrestrial animals, but accumulation happens in case of especially shellfishes, which are all filter feeders. They hold a lot of microbes, and because they don't have immune system, they don't react. They hold the microbes, and that is where the contamination is a bigger problem in case of shellfishes. Do they shed? The shedding of pathogen population, because fecal matter is actually excluded in case of land animal, whereas in case of fishes, it remains in the environment and they have chances to pollute and they can contaminate other fishes. So this is what is the major differences, where the risk will be again disposed and can be seen or can be studied in a different way. And this is what I would be talking about. So today I have framed my talk in a way like, basically I will talk about what are the emerging pathogens in the aquatic side. Maybe you will be familiar with some of them, but many of them are very, very uncommon to people who are working on animal sciences, land terrestrial animal
मैंने
sciences.
बता दिया कि भाई ऐसा नहीं, उसी की गलती है,
Oh,
लेकिन
please,
क्या
please, participants, please be muted.
Yeah,
Yes, sir. Please.
thank you.
Please go ahead.
Thank you. So, what are the emerging issues? So, you all know about new pathogens. New pathogens means which have not been recognized as pathogens and known to be pathogen now. Then new variants of known pathogens. So, you have virotype, you have serotype, you have pathotype. That they can be different at different times. So, if you do not recognize them rightly, then you will not be able to treat them or we will not be able to control them. Known pathogens, but they are found in new environment. That are also chances. They are known pathogen, but they are discovered elsewhere. So viable and non-culturable bacteria, this was a concept that was running from 1980 to 2002 or 3 or even further, where we felt that the gram-negative bacteria, they go into viable non-culturable phase and they survive in the environment. And once the trigger, a temperature trigger or any environmental trigger comes, they come back to life. So they say just like a sporulation in gram positive, gram negative could have viable non-culturable phase. Then enhanced tolerance to antibiotic and stresses. Nowadays, gradually all these microbes are getting more and more resistant, and we find a lot of new resistant, very highly resistant bacteria. That apart, other risk factors which are all bacterially produced, histamine, which is a toxin, shellfish toxin that is not bacterially produced, but there are microalgae which are all very unique to shellfishes, that is a marine environment. They could be very, very toxic, and if you eat couple of clams or mussels or bivalves, you are dead there. They could be that toxic. There are also issues of fungal toxins. So these are all major issues, the emerging issues. So new pathogens, when we say, just we saw COVID-19, even though coronaviruses were there, the one variant causes so much. So that could be different kinds of viruses which have not been known earlier to be present in aquatic environment, and they have been found to be present nowadays. Microaerophils, there no much research is going on microaerophil across the world, because it's very difficult to grow them. So usually we grow aerobic bacteria. We are very smart on working on aerobic bacteria. Microaerophilic, they need reduced oxygen to about 5% or 10%. Then they need enhanced oxygen, carbon dioxide concentration. So it's very difficult to grow. But if you look at our gut, you have a carbonate system, you have carbon dioxide, you have low oxygen, and they can survive easily. You have another group of microorganisms which are all archaea. Archaea was earlier archaebacteria, but now it is grouped as separate domain archaea. We do not know about the risk associated with them, but in many of the fermented, salted seafood, we find heavy number of those. They even outnumber the bacteria. So this is where is a cause of concern. We are still studying them. New pathotype, virotype, of course, the examples of that, non-tuberculosis mycobacteria. It was initially around 50 different types. Now it has crossed 140 types. Non-typhoidal salmonella also have been found to be causing disease in human. So if you look at OIE listed pathogens of aquatic animals, you will realize that all of them are pathogenic to fishes, mollusks, and crustaceans, but they are not passing or coming back to human beings. So this is not a big concern for us, but whenever we are giving live animals, fishes, we are exporting live fishes for commercial culture, or seeds we are sending for cultural commercial this thing culture, or we are sending aquarium fishes on trade to Singapore or other countries, then we need to routinely check all these pathogens, aquatic animals, but they are not zoonotic, not coming back to human. Today I'll restrict myself to only to food related pathogen and which could be harmful to human. So I just, so you see, look at this. This is pretty old one. Now, if you see the domains of life, you have three domains of life. You have eukarya, archaea, and eubacteria. So eubacteria will be bacteria, and archaea is separate domain, and eukarya is animal, plants, and all. So today, if you see cyanobacteria has been already grouped as microbes, and blue-green algae earlier we used to call, and they, some of them are very, very toxic. In ponds and all, they produce so much of toxins, they can kill fishes, and they can also cause a lot of skin issues to human. But epsilon proteobacteria, I was talking about microaerophils, they all fall under epsilon proteobacteria. Gamma proteobacteria are our common Escherichia coli, Salmonella, Shigella, and you have Vibrios, Vibrionaceae, Enterobacteriaceae. Everything is part of gamma proteobacteria. That we will see. Some of them are very, very harmful, and you know there are new trends. We are finding more what is that called, virulent gene to be present in some of the gamma proteobacteria, and the recent finding is that some of the bacteriophages they carry, so pathogenesis island from one bacteria to another bacteria, making them virulent. So there are many things that are happening in the field of microbiology. So you all would be familiar with Clostridium and Staphylococcus and Listeria. They're all gram-positive. They produce toxins, especially Clostridium and Staphylococcus. Clostridium produces in, say, for example, canned food, being anaerobic, and Staphylococcus, of course, there are several kinds of toxins they produce. And these are all very common in fishes as well. Gram-negative bacteria, the gram which we put them under gamma proteobacteria, you see Enterobacteriaceae is the most common food-related pathogen, but they also find their way into water. They become resident in water. They not only become resident, they also contaminate food and come back to human. So any fecal matter that we are sending, so fecal matter which is drained into the sea and water, you know, in one way or the other way, contaminate our food. So we basically remove gut, we wash them, and meat portion is usually sterile. We try to harvest them and we try to eat them. But if you imagine a city like Mumbai, the load of fecal matter. So consider one person on an average defecate about 100 grams. You can imagine 2 crore people, 100 grams. It would be means tons of sewage, tons of, I think, fecal matter. Out of that, if you see about 60 to 80% getting treated, but most of it is not getting treated and getting into the water. So that is a cause of concern. So you have mostly under gram-negative bacteria, which are all human enteric pathogens. You will find enterobacterials and Vibrionaceae, as well as you photobacteria. These are all more common gram-negative bacteria in water system. So Epsilon proteobacteria, you have read about maybe Campylobacter, but Archobacter is gaining importance now, and our group is working on Archobacter. I'll talk about that. Mostly, I'll show some of our evidences from our paper, so that will at least make you feel that somebody in aquatic sciences, in the field of fisheries, working on these areas. So we work on some of the viruses, and we have shown that viral risk being present in high risk is present in some of the seafood. And basically, I am presenting some of the evidences that is from our work. So it's not to show that we have done so much work. It is rather to draw your attention that the percentage of the degree prevalence, the prevalence is very, very high. So this is what would be my point. So a little bit we'll go into the food safety system, because everything is related food. So whether you talk about One Health in any way, environment to feed to the animals, it would come back to human as food. So you will see the basic purpose is to improve public health. So means in general, if someone is sick, it should not get moved to another person so easily. So if it is coming to the water, then there is a fair chance that it would come back to another human being. So then second thing is, if you have a good food safety system, then consumer confidence on the food supply is more. Suppose you always are afraid of eating contaminated food, just like we sometimes feel that we should not be eating street food, the way they cook and all. But when you go to the hotel, you have a different kind of confidence. So this is why the food safety system, if it was prevalent, then probably we wouldn't have that fear. Then we also work, food safety system also should be established for a sound regulatory foundation. I'll talk about regulatory foundation, how countries regulate and how we do in fisheries. So let's bit on that. So basically, if regulatory foundation is good, then you have a proper trade, then you earn money, overall development happens. So international framework. So if you look at internationally, whether do we control this thing, what it says, regulations governing international trade in food should be based on science and risk assessment. So without scientific evidence, so this is why this kind of workshop, the training program that you have is very, very important to disseminate knowledge and to know where the capabilities are lying, so that always there is a chance of collaboration, always there is a chance of exchanging knowledge. So this is why it's very, very important. So regulations governing international trade on food should be based on science and risk assessment. Risk assessment is a big area. Maybe I'm not sure if somebody has spoken, but very briefly I'll go through risk assessment in this presentation. Then SPS agreement, the sanitary and phytosanitary agreement under that. So to take legitimate measure to protect the life and health of consumer, provided such measures are justified scientifically. Again, and do not unnecessarily impede trade. I was part of one of the ISO meeting. There was a kind of imposition that all the containers of fish that has to be sent to Europe has to be having RFID tag. I'm talking about 2006-7. So, during that also they developed RFID tag. That time in India, we were not making any RFID tag. That means to say every container we are sending, we have to buy RFID tag, and we have to establish the machinery all along the supply chain to know that, okay, this food has moved from this place to this place and has been there for this long, then it has been loaded at this particular temperature, everything is tagged. So that was not possible. Of course, India voted against that. But then by now, it has become a common practice because many countries are capable of handling that. Then Article 5 of the SPS agreement, it says that, so ensure that sanitary and phytosanitary measures are based on assessment of risk to human, animal, plant life or health, taking into account of risk assessment. So basically they are suggesting that you have to do a risk assessment for every type of food that you are going to trade. So based on this kind of framework, India also launched risk assessment project from 2005 to 10. I think I was part of that. So with fish, whether it is kind of chemical risk or microbial risk, we ran the project for 5 years, and it is basically the data generation and to say that our this food is risky, this food is not risky, this food is laden with this kind of microbes, so they have to be processed in a different way, and all those information comes out from risk assessment. So let's look at what is risk assessment. Components of risk, basically, if you look at the FAO documents, you will realize that they put a word called as risk analysis. Risk analysis means you have to have management, you have to have communication. That means you are analyzing risk. Not only you are assessing the risk, you are also planning what kind of management you are going to take. And those risks which you cannot manage, that have to be communicated. Say, for example, in many food you will find it carries say MSG. Okay, so MSG, monosodium glutamate. So monosodium glutamate, some people are allergic to that. So monosodium glutamate, if they are communicating it to you, that means it's your choice whether you are able to eat or not. So that's a kind of communication. So basically policy option will come under management. Suppose you are finding risk, what you are supposed to do next. So that is part of your management. So all of you must be aware about hazard versus risk. Hazard is just anything, any potential risk, potential harm any product can cause. So HACCP, if you have read, then you will have biological, chemical and physical hazards. But risk is when there is a actual probability associated with the hazard. Means suppose say for example, is traveling in the plane is harmful? Is it risky? Uh, the person who doesn't travel, it is not a risk at all. Or the person who is a vegetarian, he is not exposed to the risk associated with, is not exposed to hazards associated with say meat or fish or egg or anything. So that basically it is kind of hazard when it is multiplied with the type of exposure, then you call them as risks. So you framework, if you see the document, so risk communication is all across, then you should have a scientific input for risk assessment, and for risk management. Also, you have a lot of exchanges. If you have assessed this is a high risk, then how you manage is different. And if you know that it's a low risk, then what you have to do, that also you know. So there has to be communication, including to the consumer. So everything is part of risk analysis. So this is a basic framework of risk assessment. So if you look at hazard identification, so hazard identification, how do you find out that, say for example, I say just now I was talking algal toxins. Algal toxins are very unlikely to come to any terrestrial animal. But fishes, when they live there, they can accumulate algal toxin, mostly aquatic algal toxin, and they accumulate there. So you can identify if you are eating a shellfish, then you know that it could be laden with the hazard could be present. That means shellfish toxin could be present. How do you characterize the hazard? You give value to the hazard that you have identified. Say, for example, say a food carrying Escherichia coli and food carrying Vibrio cholera type 1, then you will always choose the E. coli is less harmful compared to Vibrio cholera type 1. Type 1 is cholera causing Vibrio cholerae. So definitely you give higher value. So hazard characterization is coming from how the historical data, the outbreaks and things like that, and the microbe that's been isolated from somewhere. So you are characterizing that kind of hazard. You are adding value or the degree to which a hazard could be harmful. So that you are adding. So it becomes a characterized hazard. The number is also characterized as that. Suppose it's you have identified Vibrio cholerae, but what number Vibrio cholerae? Maybe or Escherichia coli. Less than 20 may not be harmful, but if it is 5 lakh, definitely it could be harmful. So the hazard characterization with respect to number and the seriousness that of disease it can cause. Now, what is exposure assessment? Exposure assessment, how much you are eating. So suppose say there are 20 microbes which is not harmful, but if you are eating a kilo, 20 microbes into maybe 1,000 times, so it becomes 2 lakh, so it becomes how much? 1,000 times, 20,000, 20,000 could be harmful. So how much food you are eating, how frequently you are eating, that becomes a kind of exposure to you. So if you multiply the how much of hazard is present and how much of exposure you have got, then you are getting a risk. Actually, the person is at risk or not. So this is what is the basic framework on which you make a risk assessment. Once you make the risk assessment, you can take a decision. Okay, this food is risky, so we have to reprocess this. This food is risky. No, we will feed it to dog or animals who are generally resistant to this particular hazard. Okay, so this is how the decision making is done. So am I still audible or I'm cut off and I'm speaking? Hello. Am
Sir,
I still audible?
yeah, you are audible, sir. Please go ahead.
Okay. Thank you. Thank you very much. So agencies which are all involved in food safety control, the international organization, of course, ISO and Codex, you all must be knowing. So they are part of international agency. In India, BIS is also giving, and Food Safety and Standards Authority also is giving different kinds of standards. So fortunately, I have been part of these standard bodies. So for time to time, so I have the knowledge what is happening in the international bank, this thing, not only for fisheries related foods. So where do we stand? So, you know, if you remember FSSAI, Food Safety and Standards Authority, and this was the bill came in 2005, then 2006 we had some amendment and it was passed when agriculture minister was Sharad Pawar, it was passed. And after that, you know, by the time it came into existence in 2010, 2011, they had their own building. By now, they are becoming strong and strong. So they have offices, they are periodically recruiting people, training experts, and you will find gradually our domestically traded food would become more and more regulated. But are we really ready to know what kind of risk our domestically traded food carries as of now? So this is where my work, means my group's work rather, is very, very important. So I will just take you through mm, some of the publications that we have where we are able to show that our domestically traded food could be very, very risky. And basically, we have to do much more to have clean water, clean fish market, and especially the type of sewage we are releasing to Mumbai waters could be very, very harmful. So are these risks real in domestic market? So are there fungal toxins? Yeah, there are fungal toxins, but mostly it is in dry and semi-dried food. Usually the fresh seafood or fresh fish, we don't find much. There are clandestine activities by some people are treating with formalin and toxic chemicals. So government has come very, very hard on those people. And nowadays we have some test kits developed and we are able to find out formalin, because otherwise they treat the fish and you will not be able to know. And parasites and cysts, they are also present. Enteric viruses are present. Heavy metals, especially ternary earlier were there in Kanpur, and gradually I think they are reducing. Gadkari is doing very good work in reducing the effect of those tannery by treating, developing a kind of mega treatment plants. Then fishes coming from polluted rivers and sewage fed fisheries. So the sewage that goes, it is treated, primary, secondary treatment, and after that fish is being cultured. So are our presumption based on scientific backing? Are we seeing that whether we are saying so safe or unsafe? Do we have a scientific backing? As scientifically it has to be proven. So this is where we have to work very hard to establish what kind of risk our food carries, and we have to test it periodically. Especially if you look at our laboratory research, 20% of fish is contaminated with salmonella. That is a zero tolerance bacteria. So when we are exporting, we don't find salmonella. That means the fish is lifted directly from inside the sea. It is washed, taken to the factory without being landed in the landing center, and it goes to the processing factory where it is washed properly, treated properly. So you don't find salmonella incidences so much in internationally treated food which is going from India. You will be surprised if you go to some fish processing factories, the EU approved and who have employed what is that called um, HSCCP and all, and you will find that they look like pharmaceutical companies. Absolutely no smell, so clean, so nice. But we are doing it for export purpose because we are getting more money. Domestic market, anything sells. We have too many people to eat, so we don't have time to really clean and prepare, and the food, the cost increases. Government is under pressure. Why the cost is increasing? So basically, we have to ensure that our domestic capability to wash, clean, and do better processing for domestic market is available to public. So close to 40% samples you find Escherichia coli. So Shiga toxin carrying Escherichia coli 25%. Then we find extended spectrum beta lactamase. Beta lactamase is the enzyme which helps bacteria to overcome the drugs, especially penicillin and the lactamase drugs. Beta lactam containing drugs. So basically, beta lactamase destroys that. So NDM is one of the beta lactamase. You see, if you remember our old stories, there was this Swedish gentleman who came for medical treatment to India because India was earlier, earlier means continuing to be cheaper medical treatment facilities. So many foreigners come. So he was contaminated with some Escherichia coli and he died there. And after that, Lancet, the journal group, there are some scientists from there. With the help of Indian scientists, they ran a program to look for the bacteria, and they traced some bacteria which were all drug-resistant bacteria, which was named as New Delhi metallo-beta lactamase. India opposed, and so many Indian scientists opposed, but they had comprehensively proven that this bacteria has originated from New Delhi. So that is why the name came as New Delhi metallo-beta lactamase producing bacteria. And you would be surprised, in our Mumbai water, we have found several such species carrying NDM. So blind NDM, we call blind NDM carrying. I'll show you the picture on that. So basically, and not only that, we are finding variant of that. So that means they become more and more resistant. So variants of NDM. The norovirus, while nobody is working on norovirus in seafood, so we found norovirus to be present in 41% of shellfishes, enterovirus is in 36% of shellfishes, and adenovirus is 21%. And Archobacter, as I said, that these are all Campylobacter related species, but they could be very hazardous. They can cause in what that called enteric diseases, and nearly 30% of the sample. And common enteric viruses, Picorna viruses, calciviridae and reoviridae. Of that, they are very important because they have low infective doses, and they have low infective doses, and they are transmitted very easily. and they cause diarrhea and self-limiting gastroenteritis. So this is why we don't care much about enteroviruses. But the developed countries, if they find enteroviruses in your food, probably they will reject the food. So this is why, kyunki the environment is so good there, and the people, once they are infected, they fall sick, they remain in hospital for many, many days. So this is how we do, basically, not only by reverse transcriptase PCR we do identify. We also do a dot blot analysis to find out that they are authentically, or mean, they are correctly identified to be the viruses. So this is one of the publications where you can just read the here that 89 samples were analyzed for RNA, 35 samples were to be positive from enteroviruses. You can see here the kind of work that we did, especially with viruses. What happened? I'm sorry, it's not moving. Okay, so another work, this is again adenovirus, 21% sample to be found to be adenovirus. Why it's not moving? Hello, am I audible?
Yes, sir. You are audible, sir.
The slide is not moving.
Okay, okay.
Just a minute. Okay. Okay, sorry.
Yes, yes. Moving.
So see norovirus. Norovirus basically, see adenovirus and other viruses are rather easily you can culture them, because the cell lines are available, human cell lines on which they can be propagated. But norovirus, we have a problem because norovirus, the cell lines are not available for norovirus. This is where it's very, very tricky. So we have hardly any publication from India on noroviruses. You see, we have found them to be in 41.3% of shellfish samples. Shellfish samples, I must tell you that they are filter feeders. So they filter a lot of water along with the food for their food. But the bacteria and viruses that come into their body, they remain there. It's a kind of bioaccumulation. And gradually what happens, if there is a phase of contamination is gone, there's a normal water, they flush out much of the bacteria. So viruses but remain. So there is a tendency that the virus will be detected, whereas the bacteria will not be detected. But this is a case in case of a area where no continuous contamination happens. So what we did, we tried to relate whether Escherichia coli and the norovirus, there is a relationship. So because if it is continuously getting contaminated with bacteria and viruses, there are likely that you will find high bacteria and high noroviruses. Unlike throughout the world, they have decided you will find viruses, you will not find bacteria. There is no correlation, but we established that there is a correlation, a strong correlation. Sorry, again, it's not moving. Okay, okay. So then coming to another big area of research that we have done. So especially Archobacter. So people who are familiar with epsilon proteobacteria may appreciate this, that there was a Helicobacter pylori which gave to Barry Marshall in 2005 a Nobel Prize. So basically they established that this Helicobacter is causing gastrointestinal ulcer. And if you see 70% of biopsy samples of gut, you will find that this bacteria to be present, Helicobacter pylori. And that is a pioneering work for 20, 30 years only on one bacteria. So they could establish that it is to be present. But when we started working, whether because it can survive, because it's also a fecal oral origin, it should be going into the sea and coming back to the food. We did not find, and accidentally we found Archobacter, and we looked for Archobacter, and now we realize that Archobacter is also as common pathogen as Salmonella. So you said they're microaerophilic, they grow with 20% carbon dioxide, 5% oxygen, they need nutritionally rich media, and they can cause severe gastroenteritis. And our paper has been also been hosted at a FAO site for this one. I don't know, this become very slow. Maybe net is slow or okay. So our investigation, if you see Archobacter buccellaris, Chiroe, and Cryophyllus, they have genetic potential and they have been found very commonly. Our publications are also for 4-5 years it is there. Then we are basically now working, developing easy diagnostic assay. I have got a DBT project on that. And another laboratory that is working on Archobacter is IVRI's Kuldeep Dhama's laboratory. So Kuldeep Dhama, by this thing, he is my training batchmate before we became scientists. So happy to know that he is working, and we have not shared much of the information yet, but then at least 2 laboratories in India working. This is a good area to work. So okay, so you see the number of publications gradually how it is increasing. So you will find in another 10 years time they to be very, very important bacteria. So this is why I said it's a emerging bacteria. So as I said, Helicobacter we could not find, Campylobacter we have not started. Well, this is a cross contaminant mostly from terrestrial animal. So we have not really worked on Campylobacter, but it's time to look at Campylobacter as well. So some of our publications on Archobacter, you can see quite very, very common, very prevalent. And we have also studied their genetic diversity in fish and coastal environment, and lot of samples are being contaminated. So it's the sample number 200, 300 samples we have analyzed, and we're still continuing to look for these things. Again, we have found Archobacter in retail markets, not only even malls, wherever the fish is kept very nicely, there also we find Archobacter. Then we have some work on, as I said, when the microbes become, they carry different kinds of genes. So then also it is considered as an emerging pathogen. So this is an emerging scenario. So you say cigatoxin toxin gene. So you have to consider that on a elevated hazard category. So that is why it is very important to understand that we have different kind of pathogen. They have analyzed in food, and there has to be a risk assessment done. So you see again, we have found 100 samples, 100% of samples are carrying Escherichia coli. And this is basically to say you that the type of water they clean the fish. So if you go to the fish market, the type of water they use for cleaning fish, and the type of handling they do, the hygienic knowledge, the knowledge on hygiene, the common people have. So you can get, say, Escherichia coli not only can come only from human fecal matter, even animal, birds, and everything would carry. So if this place is not clean, then you know definitely you find the access of bacteria to fish. Similarly, survival behavior of Salmonella, we have also shown that Salmonella can survive in this paper 90 days at minus 18 degrees centigrade. So if you ask me, Escherichia coli, if they get washed into the sea, whether they survive there, I would say they survive there. Very much they survive there. Escherichia coli can survive. My study shows that at least 2 years in seawater. They may lose their virulent characters, but the Escherichia coli can survive for more than 2 years in seawater. I studied in artificial seawater. They can survive 2 years. Similarly, you just saw the Salmonella is staying for 18
Sir, you are not audible. Hello, sir. Point five प्रमाणे करावं. Hello, Naik sir, you are not audible. ते sanction घ्यावं repeat घ्यावं लागेल ना. Hello, sir, you are not audible, sir. Naik sir. You can log in again and then you can try, sir. Yeah.
Presence of New Delhi metallo-beta-lactamase in seafood in quite high number. So archaea are very common in salted food. In salted food, we are finding the dominance of archaea over bacteria. So we are unable to assess the risk associated with archaea. And this is the dominance. You see, orange line is bacteria. Now it is still visible.
Yes, PPT is not visible now, but you are audible and visible.
Is this okay now? Visible also?
Yeah, you are visible, audible also. Please share your PPT, sir.
Okay. Maybe there were. Okay.
Please continue.
Yeah, you can see how much of bacteria are present and how much of archaea are present. The blue line is archaea, how much of bacteria. You can see the proportion of bacteria and proportion of archaea. So in salted and fermented food, you have more of archaea rather than bacteria. So this is what is the highlight of this one. And why we are worried about archaeal presence? Because we do not know anything about that, whether they produce toxin, whether they are capable of producing histamine, what kind of, whether they can ever cause disease. But unfortunately, we cannot remove them also. So if you see, we tried several ways, heat and other things from fermented foods. You see, even with 6 kilo gray, 6 kilo gray is very, very high for any food to be exposed to. And still you find those archaea to be surviving there. So this is what is actually our worries, and our work continues on that line. We collaborated with BARC on this. So anyhow, we have not yet published, but then we will be anyway looking at that. The summary of my presentation is that high incidences of emerging pathogens are there. So high load of archaea with unknown risk factor. Antibiotic resistant bacteria are also present, which are all getting from hospital waste directly into the water, and they have been detected. So we are also finding high load of viruses that are in shellfishes. So it's time to do more and more of risk assessment studies and domestically marketed fish. Then what is need to be done? So risk assessment projects, pan India and long term. We have to link hospitals and R&D institutes because always our work stops only at that. At what our analysis is the end of our work. But if we link to the hospital, then we can always risk it to the we can connect is available, which food carries what kind of a, or whether it is actually causing disease or not. So this is why it is important. So the BIS and FSCCI also should be linked to R&D institutes to get input. And we have to also develop general awareness about the risk associated with fish and fish products. Thank you very much.
Thank you very much, sir.
Sorry for that network glitch, which took over 5-7 minutes.
Yeah, thank you very much, sir, for your valuable information and the emerging safety issues in aquatic foods you have highlighted. Very excellent talk, and that will definitely help for the audience, particularly PG and PhD students. Thank you. Then I request the participants, you can ask the questions. Participants, please.
Yeah, there is a question, is microplastic hazard is a concern in fish and shellfish products? Yes, it is a concern because now a lot of work, we have some good work on microplastic. My colleague actually, Dr. Martin is doing on that. But then we have been finding high incidences of microplastic. And the way we are using plastics, and we throw everything into the water, and it's no doubt that they will find their way. They will break down into small bits and become part of the fish tissue. So yes, it is a risk because some of the plastics will release some toxic metals into the fish tissue. That is how it is risky. It is environmentally risky, yes. Plastics are generally environmental risk, but microplastic, we do not know too much about them. But then we are studying at present that whether they can release the toxicant, colorants, into the fish muscle. Is there antibiotic AMR seen in aquatic animals? Yeah, our NDM work, my colleague Dr. Sanat Kumar, he's doing excellent work on this antibiotic resistances. So you find we have found multi-drug resistant bacteria, mostly Enterobacteriaceae, plenty of them, and this is where I talked about extended spectrum beta lactamase and as well as I said NDM on that. So we find we are running a project on AMR. Maybe we'll continue. That project is over last year, but it is supposed to continue again. Most common pathogens found in aquatic animals. If you ask me common pathogens in freshwater side, you find aeromonads. See, only one Vibrio in terrestrial side you may know, that is Vibrio cholerae. So Vibrio cholerae is considered to have originated from aquatic water, and it has become pathogenic. But there are several, about 30, 40 species of Vibrios available in the ocean side, marine side. And one of them, many of them are also luminescent, and many of them are pathogenic to fish, shellfish, shellfishes. And some of them are to human also. So especially gastrointestinal diseases they cause. There is one question, research topic on fish and aquaculture related hygiene and safety. Hygiene, see the work that I presented today about Archobacter, Archaea, very difficult to do. They need specialized conditions. So if you want to study on hygiene and safety, you have to track maybe a simple organism, maybe a SHA coli or one, the presence, dominance at different levels. So how it is coming from landing center, whether it is coming from the handlers, whether it is coming from basket, from what is the source. So that kind of studies can be done. You can study survivability of, you can have some pathogen and study how long they can survive against some, what is that called, say, unfavorable condition. So that would show that if environmental condition or processing conditions are there also, they can survive. Can we consider One Health concept for aquatic animal? Yes, much of the consumption is water. So if you see whether you live in an environment where 70% is water, so definitely the weather, it is microbes going into water or coming out of water, you are always linked. So One Health concept, of course, is like a mega approach, ecosystem approach. So you are looking at everything together. Yes, definitely they are related. So is it a fact that the fish get infected during the month of Kartik, for which it is not eaten? I know in side like Odisha and West Bengal, Kartik is considered to be, that has got been related to, in fact, environment. So mostly some fishes have got single modal breeding cycle, and some have got two breeding cycle. So there is nothing to prove that in Kartik they get infected, so it is not eaten. Rather, if you don't eat in Kartik, you allow the fish to breed. So that is a more possible explanation. So the question was, in fact, is it a fact that the fish get infected during month of Kartik, for which it is not eaten? It's not true. We have no evidence to show that. Usually, it is related to for the fishes which are got one breeding season there. So I cannot answer. It's difficult for me to give a reason for that, but then I would suspect that it is more likely to be related to breeding pattern of some of the dominant fishes there. Am I audible?
Yeah. Yes, sir. You are audible. I think. Is there any question? Last question. Okay. So thank you once again, sir,
Okay.
for
Thank you. Thank you very
all
much.
these
Pleasure talking to each one of them.
yeah, important issues related to aquatic foods, and you highlighted about the emerging pathogens, including viruses also. Thank you once again. I request all the participants, please start your video for group photo, please. For group photo, yeah. Yeah. Thank you. Now, after this session, there will be validatory program. So, yeah, kindly wait, all the participants. We will have
Okay.
some information also. Yeah.
Okay. Thank you very much. Can I log out?
Yes, sir. Thank you very much, sir. Yeah. So now for the validatory program. Once again, good afternoon to all these dignitaries for the validatory program. We have with us our course director, PI of Nayab Caste MAPSU project, and Professor and Head Department of Veterinary Public Health, Dr. R.J. Zhende, sir. So, sir will act as a president of today's validatory program, and he will give the concluding remark at the end. Today, we have with us uh, Dr. S.D. Ingole, sir. He is working as professor and head department of veterinary physiology, and also he is the nodal officer for international training, as well as he is the co-PI of Nayab Caste MAPSO project. So, and then Dr. Vivek Shukla, he is associate professor and sectional head department of livestock products technology. He will give the vote of thanks. And so, dear participants, first, uh, thank you very much because of your all cooperation and support. Now we have come to the at the end of this 4 week certificate course training program on enhancing livestock production and food safety through One Health approach. So now I request Dr. S.D. Ingole sir to say few words, brief. Okay. So first, I request the one or two participants for your feedback, for your suggestions. Yeah, your opinion about this 4 week certificate course program. So now, any participants, you can give your comments, please. Yeah, you can speak. Participants, please, you can, yeah, speak.
Please
Yeah.
unmute yourself and speak. Please introduce yourself first, and then you can say whatever your opinion about this 4 week training program we are conducting so far. Okay. You were just we are providing the feedback form to everyone, and also post evaluation form also to everyone. Okay, but on behalf of all the participants, any one or two person can speak. You are allowed. Okay, please unmute yourself and speak and introduce yourself first.
Very good afternoon, sir. I am Dr. Gopal Patra from Department of LPT, West Bengal University of Animal and Fishery Sciences, Kolkata, sir.
Okay.
So first of all, I would like to thanks to the Jande sir and Vaidya sir and all of the organizing member for selecting such type of nice topic, because all of we know that in coming days it's a very great issue, because we are going to production, engaging the production. So not only production, qualitative production. So for qualitative production, so we must be concentrated about the food safety issue. So that's why, and also I want to like to thank for selecting a good number of eminent speaker. They have given their best lectures, I think, as per my knowledge. So I am very much enlightened and enriched also. So I would like to request you, sir, if possible, sir, if you kindly provide us a soft copy of the write-up, or if possible, sir, PDF copy of PPT. So I will be very much benefited, sir. So thank you all, sir.
Okay, okay. So about all this PPT in the manual we are preparing, the Dr. Vaidya will speak about that later on. Okay. Thank you. Anybody else?
Hello.
Yeah.
Yeah, yes, madam.
Introduce yourself.
Yeah, good morning, everybody. This is Dr. Sonia Chavan. I'm basically a surgeon for last 34 years. So basically, I had seen this One Health program from the point of animal welfare issue. So there is so much you are given as from different topics which are covered, and there's so many people which are involved in strengthening the safety and livestock production issue. So there are different aspects of the same One Health program. That is what I came to know, and it will definitely help us in our future. Thanks a lot. It was really good, and thanks for the organizing committee for organizing such a well-planned program. Thank you.
Thank you very much, madam, for your suggestion and good words for the program.
Sir,
No, yes, yes,
uh, is it I'm audible, sir?
yes, yes, you are audible.
Sir, myself, Dr. Rajesh, working as assistant professor in the Department of Livestock Products Technology, Gadwasu, Ludhiana. But presently, I am at South Korea, sir, for international training. And even though in South Korea, I am regularly following all the lectures. And first of all, I would like to congratulate the. Yes, sir.
Very good, Rajesh. Yeah.
First of all, I would like to congratulate the PIs and all the co-PIs for choosing this topic. So, in spite of my busy schedules, but the topics which you have chosen, sir, are like very interesting one. So, I think this is like fruitful outcome from this training program. So, thank you very much, sir.
Good, very good. All the best, Rajesh, for your training program over there.
Yes, sir.
And
Yes, sir.
the best. Okay, thank you.
Thank you, Dr. Rajesh. Any other participant who want to speak? Okay, okay. Thank you. Thank you, all of you. Now I request Dr. Ingole sir to say a few words about this training program.
Thank you, Dr. Vaidya. Good afternoon to all of you. So we are now almost at the fag end of this training program, 4-week certificate course, which was on enhancing livestock production and food safety through One Health approach. So overall, this 4 weeks we had a good amount of participants, and I think almost everyone was we.
I request the participants, please be muted.
So I hope that all the participants must have been completely benefited with this course with the following objectives that what we have thought regarding this course, which will help us to what are the challenges in the AMR and explore some new innovative approaches for a safer food production. So, in respect to that, as we know that the food security area, and recently, if we think of yesterday only, what we have seen, the hunger index also. So, with regards to that also, what this food security will be helpful to us to improve our hunger index of our country. So that is also one of the important issue that we have to look into. And besides that, so whatever the speakers that we had, so like almost about 40 speakers we had, we tried our level best to give you the best out of it so that it will be beneficial, and you can in future, like all the participants can work on to it, and that too in the area of One Health approach in the livestock. So I wish all the best to all of you and the best wishes to the participants. Thank you.
Thank you, sir, for highlighting the overview of this 4-week certificate course program. Now, I request Dr. R.J. Jhende, sir, who is the PI of this NAIPCAST MAPSU project and the course director of this training program, to give the concluding remarks. Jhende, sir, please.
Okay. Thank you, Dr. Vaidya and all my team here. Uh, Today we come to the end of this hectic 4-week certificate course on enhancing the livestock production and the food safety through One Health approach. And I would like to inform you that the kind of support we get it from our university authorities and our college authorities, associate dean, for organizing such programs for enriching the knowledge of the today's youth, uh, of the PG, PhD students and the faculties working in the different veterinary colleges across the country. So with this intention, we have been organizing such training programs under this NAIA project, which is World Bank sponsored project for the Center of Advanced Agriculture Science and Technology. And this will be in future also, we will be organizing such a 4-week certificate course. 2, 3 more courses will be coming up in next few months. That was very great, the response for this kind of the program. There are about 480 participants were registered, but every day we got on an average, the attendance was 195 to 225. So there was very rich experience of attending this kind of the participants, and they getting the knowledge in the various aspects of the food. And this is the training program in which we have covered all the 3 aspects, that is food safety, food security, and the One Health approach, including the AMR, what today's burning topic in our either human as well as animal population. So I thank my organizing team for getting such all experienced people in their own expertise area, for contributing their own experience and the latest informations available in their particular research area, or their food safety area. So I take this opportunity to thank all the speakers for making their available time over there in this training program, and delivering the very good talk of their own interest. Okay, and I'm definitely happy for getting this enrich the knowledge of the all the participants in the different aspects of the food safety, animal food safety, and definitely it will help in their day-to-day life, and also designing the different kind of the that research areas for their future planning. So with these few words, I conclude my speech here, and then once again thank my authorities, university authorities, and also the college authorities for being and encouraging us to organize such training programs. Thank you so much.
Thank you very much, sir, for your presidential address and concluding remarks, and highlighting about the importance and the of the training program and benefits to all the participants. Thank you once again for your wholehearted supports and also guidance to all the organizing team for this organizing such a wonderful program. Now, one announcement before the vote of thanks. There is announcement. All the participants we have mentioned in the chat box that two links we have shared. First link is for post evaluation pro forma. So you have to fill up the evaluation form, and another is feedback form is also we have shared in the chat box. So that you have to fill up and you have to submit. And another that we have shared the bank details also. Those who wants the certificate, those who have the more than 80% attendance, that we will see. And then those who want the certificate, means after the post evaluation of your whatever the marks you have achieved, and you have passed the this based on the attendance, and total evaluation will be there. Based on that, we will provide the certificates. But for that, you have to pay the fees. For the staff, means faculties and working personnel, you have to pay 500 rupees. And for students, students has to pay 300 rupees. So that details are there in the UN browser, and we have shared in the chat box so that you can see. So now I request Dr. Vivek Shukla, co-PI of this NIAPCAS project, to propose the vote of thanks. Dr. Vivek.
Thank you, sir. So good afternoon to all the participants. So now this 4-week training program on enhancing livestock production and food safety through One Health approach has come to an end. So I take this opportunity to propose the vote of thanks for this training program. So first and foremost, I would like to thank from the bottom of my heart from all of our team. I would like to thank our Honorable Vice Chancellor and Colonel Dr. Professor A.M. Paturkar sir, who is the main striving force for organizing such a long duration training program. So thank you, sir. Thank you for your support and guidance, without which it was impossible to arrange such a long and nice training program. Then I would like to take this opportunity to thank DDG Agriculture Education and National Director NAHIP ICR New Delhi, Honorable Dr. R.C. Agrawal sir, for granting such a nice project to our university and our college, and for giving permission for such training program. Then I would like to thank Director of Instruction and Dean MAPSU, Dr. S.V. Upadhyay sir, respected Dr. S.V. Upadhyay sir, for his guidance and encouraging us to arrange such training program. Then I will take this opportunity to thank our Director of Research, respected Dr. N.V. Kurkure sir, for his encouragement and always promoting us to organize such a training program for the benefit of the students as well as the faculties. Then I would like to thank our beloved Associate Dean, Dr. Professor A.S. Ranade sir, who is always the constant source of inspiration for all of us and always guiding us in arranging such training programs. Then I would like to thank other authorities of the NAHIP for granting the training program. Then I would like to thank Dr. Jhende sir, Dr. Gaikwad sir, Dr. Ingole sir, Dr. Vaidya sir, Dr. Patil madam, Dr. Mishram sir, and all other co-PIs involved in this training program for giving their time, for giving their precious time to make this program a success. Then I would like to thank Dr. Rajpal, Dr. Anu, and Ashwini, who are taking the lot of pain in arranging this program and making this program a grand success. Without them, this it was impossible to arrange the program in such a nice and fruitful manner. Then I would like to thank all the staff members of the NAHIP, Mumbai Veterinary College, and and all the staff members of the Mumbai Veterinary College who have taken all the efforts to make this program a success. I would like to take this opportunity to thank all the speakers who have delivered who have delivered the nice lecture. They have given a lot of time for preparing such a good PowerPoint presentation. They have shared the write up also. So I would like to thank all the speakers who agreed to us in giving their time and delivered their lecture during this training program. Then in the last, I would like to thank all the participants who were constantly joining the program, who are the source of inspiration for us in arranging such a program. Because of without you people, it was impossible for us to arrange such a program, because participants, they are the most important in any event. So thank you to all the participants. And in the last, I would like to thank all the team of NAHEP, all the staff members who have helped and supported us in arranging such a program. Thank you one and all.
Yeah, thank you, Dr. Vivek. Now one more announcement. We have shared the YouTube link. So those miss all participants, those who wants the PPT, so they can. Yeah, we have shared. You can see that you can use that YouTube link also. All the lectures are there, and we are preparing the compendium. So those who will get the certificate, and we will send the compendium to them also. So now,
Sir, please share all links in the WhatsApp group.
yes, sir, yes, sir.
All
Yeah.
links in WhatsApp group.
Yes, yes. we will
Because
share.
it will be out now. So all links should be sent in WhatsApp group.
Yeah. Hmm. Yeah. So once again, from the organizing committee, we are requesting you. You have to fill up the post evaluation pro forma, then feedback form, and then those who will pass in that pro forma, you will get the certificates. But for certificates, you have to pay, as I told, 500 rupees for the faculties, means working personnel, and 300 rupees for the students. You will get the certificates. Yeah. So already we have shared in the chat box account details, and that also we will share in the WhatsApp group also. Okay.
It will be sent, post evaluation sheet will be sent within which date? Today? Instant?
Yeah, different links are there. So yeah, and today's